Welcome, everyone. Thank you for joining us for our presentation titled Extraction-Free Sensitive Detection of Viral RNA Using Direct RT-PCR Chemistry. It's my pleasure to introduce you to Donald Green. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our presenter will address your questions following at the end of the presentation. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Donald Green. I will now turn the presentation over to him.
Hello, everyone.
Hello and welcome to Lab Week. Thank you, LabRoots, for giving Cytiva the opportunity to present. My name is Donald Green. I am a field application specialist for Cytiva. And today I will be discussing our new direct RT-PCR kit. The kit can be broken into two parts, the AIC mix, which is the part that makes it extraction free, and the RT-PCR chemistry, which is also Lyell stable. I'll get into more of that later on in the discussion, but please, I hope you enjoy the content, and I look forward to you reaching out with questions. Before we jump headfirst into the kit, I will discuss a few other topics. The first one, the PCR and RTPCR market. Polymerase chain reaction, or PCR, is one of the major advances in the field of molecular biology, used in diagnostic and research laboratories the world over, to generate really sufficient quantities of DNA to be tested. The global reach of digital PCR and qPCR has been expected to grow by leaps and bounds. The increase is really due to the availability and higher demand for different kinds of master mixes for the technology, government funding to the diagnostic and life science sector, and really an evolution of PCR variants that have led to an increased application in the diagnosis of infectious diseases. The next question would be, why reverse transcription quantitative PCR or RT-qPCR? We use RT-qPCR to amplify a transcribed cDNA from RNA into multiple copies. It's quantitative, which means it measures DNA concentration in real time, and it typically has a one-step process or a two-step process. In the one-step process, we use it for large samples to really reduce chances of the contamination process in the process. The two-step process is preferred when sample is scarce or multiple sampling may be required from one sample. So if we take a look at where RT-qPCR is widely used, we can see that it's been widely used to calculate RNA expression levels within tissue, diagnose genetic disorders such as muscular dystrophy, or diagnose diseases, certain viruses which have RNA genomes such as, you guessed it, COVID-19, HIV, HCV, etc. So next we're going to take a look at the global market for PCR technology by type, focusing in on the RT-PCR market share. The market share has been around 13.7% for the past few years, and it was expected to grow on average at around 5.2% between the forecast period, which was 2016 through 2021. As you can imagine, that number has probably been blown out of the water currently, but I haven't seen the updated numbers. But as you can imagine, everyone is really using RT-PCR, qPCR for the COVID-19 pandemic. But previous to the pandemic, why was this technology expected to continue to grow? Well, the increase of testing viral pathogenic agents had already started. The technology also obviously gives us the ability to convert RNA into DNA, which is very useful in the diagnosis of infectious diseases, i.e. COVID-19. And the sensitivity, the accuracy, and the reproducibility, which was being processed and has been gained severe importance in the past decade just due to its capacity for making complementary DNA. One of the last things I'd just like to review is really the difference between extraction versus direct applications. So if we take a look at the application area, nucleic acid amplification testing is preferred over traditional culture techniques due to higher sensitivity and speed. It's used very widely in applied market testing to detect infectious diseases and metagenomics, and I'm sure you can list out some of your own. What are some of the challenges and opportunities associated with just doing RNA extraction? Typically, some of the challenges in RNA extraction include the barrier to scale up the testing in the molecular pathology lab because of lengthy extraction steps, because of complex protocols, because of the number of reagents required, because of the cost, the expertise needed with hands-on, and then the compromised sensitivity with some of the older methods. Where extraction-free methods come into play and give you opportunities, they can be used as a screening tool in high throughput and scale up setup where time to result is critical. They reduce the extraction kit bottlenecks, which have been particularly prevalent with the current COVID-19 pandemic, and they provide a simpler, more cost-effective protocol, as well as eliminate cross-contamination risks during extraction setup. And what we can say is the nucleic acid yield is usually not as variable as it is with the extraction process. So if we take a look, just an overall high-level viewpoint of traditional extraction versus our direct RT-PCR approaches, traditional extraction is still great, right? It's the gold standard in traditional method, gives you high levels of purity. You can use the aliquots later for different tests, save the rest. Potential for larger samples and gives you very high levels of concentration. However, for the direct chemistry, if you're looking for a quick TAT or rapid turnaround time, it gives you a rapid protocol, fewer steps, less time, less hands-on time, which means less contamination potential. Very cost-effective, convenient, one vial containing all the critical components for the lysis, and it handles small samples with very good sensitivity. Thank you very much for listening to all the background information, giving us a greater foothold on understanding the purpose for our RT-qPCR chemistry and our direct RT-qPCR kits. So what is the Cytiva Direct RT-qPCR kit? Our kit can be split into really two main components. We have our AIC mix, which is our anti-inhibitor complex mix, which really protects your RNA. That's the secret sauce, if you will. And then our master mix and reconstitution buffers, which is basically our qPCR master mix dried down into a lyophilized cake, and then our reconstitution buffer. You combine both of those, and you get our direct RT-qPCR kit, which gives you the robust extraction-free chemistry that has been optimized for a one-step detection of RNA, really optimized towards detection of COVID. Continuing in the vein of what is the Cytiva Direct RT-qPCR kit, what we want you to really take heed to is this kit allows a user to really lyse the sample basically by heat, with the AIC mix stabilizing the RNA against nucleases and PCR inhibitors. Just an aliquot of the lysate is needed, and you can add it directly into your RT-qPCR master mix, providing a highly sensitive specific amplification and detection of RNA from various sample material in less time. And when I say less time, if we go over and look at the figure to the right, you will see if you're taking from step one of the gray boxes, you collect your sample, then you go into RNA extraction. We say RNA extraction is approximately 60 minutes because that's really going to depend on the kit. It's going to depend on the skill of the person performing, using the kit, and it's going to really depend on, a few other factors, how far things are apart in the lab. If you come down to the direct RT-PCR kit, you will see that taking your collection of your sample goes directly into your AIC mix. We say approximately 10 minutes, but it's 5 to 10 minutes. And what you can see is the amount of time you save from the green box and the gray box during what we would call the extraction free versus the RNA extraction. That's where you really get your time savings. You can imagine if you're doing this extraction 10 times a week, just look at the amount of time you would save over the course of the week. So I really want to hammer this home to you in a very feature benefit setup. So one of the features, again, you lyse the sample by heat. You put your sample directly into the AIC mix. The AIC mix is a very good stabilizer of the RNA. You heat it for 5 minutes, and then you take an aliquot of that lysate and directly into the QRT master mix. So it's a very streamlined, easy workflow, time and cost savings, minimal handling issues, and minimal sample loss. Our kit includes all the critical reagents for sample to result workflow. There's a stabilized master mix available in cake or bead format, which really provides you a very ease of use, convenience, very automatable. Some of the benefits that we'll discuss in the data down below, you will see that with our kit, you actually have a higher tolerance to transport media, which allows you to be more capable of detecting low viral loads, even when the sample transport media volume reaches around 45% of the final reaction volume. And you also get increased RNA stability of the sample. And we've been shown to detect COVID-19 particles down to approximately 10 copies per reaction. So the benefit there is the kit is very robust, sensitive, and very reproducible. Lastly, the flexibility of the kit. It works with a wide range of sample types, swabs, cells, blood, and body fluids, compatible with both real-time and endpoint RT-PCR, compatible with a wide ranges of transport media, and suitable for manual and automated protocols. Really quickly, before we go into the performance data, I want to give some background into our actual genomics portfolio. It's very relevant with a wide variety of different customer market space. Future molecular diagnostic applications fits in with qPCR, NGS, and other types of nucleic acid testing. We have some great DNA isolation kits, our Sera Extractor cell-free kit, which minimizes genomic DNA, our Sera Extractor genomic DNA kit, which minimizes RNA within your sample, our Sera Extractor virus pathogen kit, which has been used very, very well with the COVID-19 pandemic. It's really designed for that. And then we have our extraction-free chemistry, which fits right in here. We also have our amplification chemistries. If you have particular amplification issues with maybe a single cell with Genomify, if you want to use Templify, which is for circular DNA, Genomify is for linear DNA. And then we have some size selection and PCR cleanup reagents, what we call our Ceramax Select, which really fits directly into your workflow and gives you one bead option for PCR cleanup and size selection. If you want to know any additional information about any of these products, please don't hesitate to reach out to me. And now we will get into the performance data. Jumping directly into our RNA detection, which is exactly what we need to discuss, right? These are the performance data that you need to see. Just a bit of the background. We'll have a bar plot, and then we'll have a standard curve plot on your right, bar plot on your left. In the bar plot, you'll see the primer set N1 targeting the N gene of COVID-19 based on CDC suggestions, obviously. The plot shows the results combined from 3 independent experiments with technical replicates run each time. The positive control primers are targeting the human RNase-P gene, which I'm sure you're very familiar with. We are also used a plasmid template control, which really is a plasmid coding for the same artificial sequence contained in the viral particles. And then we did an artificial viral containing a portion of the COVID-19 viral genome in that last portion. And what you can see here is we wanted you to see the difference in the protective capabilities of our AIC mix versus no AIC mix. You can see in the first bar, the far left, we ran the samples without any AIC mix for the N1 primer set as well as the RNase-P. You can see without any AIC mix, there was no protection, so there were no results. Basically, as a failed experiment. Simply put, putting it with the AIC mix for both the N1 and the RNase-P, you will see that the experiment passed based on the CT value of around 35 or so. And RNase-P, relatively low because it comes up with basically anything if you've used it before. It's very, very robust. We all know that. And then again, we tested it with some artificial primer sets as well, the plasma template control and the artificial N1 virus. And you can still see with the AIC mix, we still get great results. We actually obtain results from just 5 minutes sample right into the AIC mix. So the next thing I want you to take a look is to your right. You will see the standard curve really demonstrating the linear and sensitive detection of viral RNA across a wide range of viral loads. If you take a look at the 10 viral particles per reaction, you'll see the N1 will still detect it around the same CT value, correct? Then you take a look at the 100 viral particle reactions, it was detected, 1,000, of course it was detected, 10,000, and so forth. The numbers on the right are not surprising. The numbers all the way to the left, at the 10 viral particles per reaction, really shows the sensitivity of our kit. Why is this important, you ask? Well, as with COVID-19, you may not have a ton of virus yet on your system. So if you're able to detect down to a very small amount of copies in the sample, you can still trigger a response that, oh, this person has COVID-19. That's very, very important. And then if you take a look at the RNase-P, we just wanted to keep that in there to let you know that the RNase-P worked fine in the experiment. So there's nothing screwy going on. The next discussion we're going to have that I mentioned earlier is the tolerance to transport medium. This particular case, we are using nasopharyngeal samples from COVID-19 negative donors, and we spiked them with heat inactivated COVID-19 viral particles. So if you take a look here, and this was very surprising for us, right? This wasn't something we started out with the expectation that it was going to happen. But if you take a look, the Sativa Direct RT-PCR chemistry exhibits very high tolerance to transport media compared to some of the others on the market. Some of the others on the market really tap out at around 25% or 30% of the transport media being in the final reaction volume. But what we can say is that with our detection, we can get up to 45% of the final volume of the transport media, and we still see benefit. We still see results. If you think about it, this can be very beneficial when testing borderline negative or positive samples. You're essentially doubling the sample volume tested without affecting the assay sensitivity. So just going over to the bar graph, you'll see that we tested at 25% and we tested at 45% for N1, N2, and then RNase-P. And you can see the results speak for themselves. They are basically the same. So this is a really great finding for us, right? And many of our customers really appreciate it because they can put in, again, more of the transport volume, which increases the probability that they actually have more of the virus on board to run their sample with. So the next experiment we wanted to carry out with the kit was using different transport media. Obviously, with the amount of people going into trying to test COVID-19, transport medias were pretty widely unavailable. The CDC actually put out a recommended alternative collection media that can be prepared very, very locally, including PBS, saline, and then the more widely used standard universal viral transport media suitable for cell culture. What we did is the graph to the left shows you that the Sativa Direct RT qPCR chemistry allows the detection of viral load in a wide dynamic range, really irrespective of the transport media it's used in. Again, we looked at PBS, saline, and then just a standard universal transport media for cell culture growth. And using positive nasopharyngeal swabs, we were still able to see the no template control. There was nothing there. And then the dilution factor of 10,000, 1,000, 100. One of the last experiments we did was really just to show that this doesn't just work with COVID-19, which is where the kit was really birthed out of, but it can be compatible with other viral particles. So we took a look at Ebola, and as similar as we did previously, we did a no AIC mix with an AIC mix, and you can see the results that even under challenging conditions really defined by nasopharyngeal sample, we can detect Ebola viral particles. Thank you for listening to all the data. I'm sure it was very exciting, and I hope you guys really appreciate it. Last thing we're going to do is take you through a quick summary of the kit, kind of hit some frequently asked questions and some of the top tips that we have available currently. Taking a look at this quick summary slide, what we're going to say is the Cytiva Direct RT-PCR Kit enables a one-step extraction-free procedure using nucleic acid amplification testing technology. It addresses a few needs here, right? It addresses the need for rapid and high throughput RNA detection, provides a very sensitive and robust reproducible solution with highlighted information such as increased tolerance of up to 45% of the universal transport media, increased RNA stability within the sample, and again, we specifically developed this kit out of the need for COVID-19. And we can show pretty successfully that we can detect COVID-19 particles down to approximately 10 copies per reaction. This full kit includes all the critical reagents, really allowing kind of a sample to, or increasing the speed of sample to result workflow. Works with a dynamic range of sample types, swabs, cells, body fluids, and it's pretty easy to set up for manual or automated protocols. So one of the things I wanted to take you through, as I mentioned, is the frequently asked questions about the kit, and hopefully we can clear up some of those for you. Can the Direct RT-PCR Kit be used with any universal transport media? The answer is yes. However, we do have issues with UTMs containing lysis reagents already, right? If the AIC mix is really helping you lyse based on heat, we don't need the sample already lysed. So any UTMs that contain lysis reagents such as SDS or guanidinium should be out of the picture. They don't work really well with the AIC mix. Can I use other sample volumes other than the ones recommended? We recommend 5 microliters of universal transport media input volume as a guide. However, based on the data we've shown you previously, the sample can be further optimized with the addition of the universal transport media volumes being increased. Being between 3 microliters and 8.5 is what we've been seeing kind of in our labs. As long as you're keeping the AIC mix as a constant 6, this is what we encourage. What sample volume should be used for the RT-PCR mix? We recommend a total of 11 microliters of lysate, meaning the 5 microliters of sample plus 6 microliters of AIC, to be added to the RT-PCR master mix to get a final reaction volume of around 20 microliters. My starting sample is old and could be degraded. What can I do? Due to the unstable nature of RNA, we recommend working with fresh samples where possible and store samples in UTM as recommended by the collection to manufacturer. Can I use the AIC mix to inactivate the virus? Cytiva has not validated the efficacy of viral inactivation at AIC within the AIC at 70 degrees. Incubation time for 5 minutes. Lab optimizing to use our formulation would need to do need to satisfy themselves that they have sufficient approvals to work with infectious agents using our kits. Can I scale up volumes recommended as it is difficult to pipette smaller volumes? We understand that, however, the accurate pipetting of the smaller volumes is recommended to really mitigate issues when working with samples with higher viscosity. The AIC mix is not working properly. What should I do? It should be considered that the efficacy of the Cytiva AIC mix always depends on the sample matrix or the type of swab solution and the RT-PCR kit to which it was used. The top tips that we have seen with customers testing our kit, using the AIC mix, we recommend mixing the AIC mix before use in the aspiration step as it tends to settle if left to stand for long periods of time, 5, 10 minutes, 15 minutes, post-mixing and on ice. When inhibition is observed with the direct RT-PCR kit, we recommend adjusting and or reducing the sample volume added to the reaction. Some RT-qPCR master mixes are not robust enough to handle the inhibitory agents present in the lysate or present in the AIC mix plus sample. So if you are just using the AIC mix alone with your in-house RT-qPCR and you're not really getting good results, where inhibition is suspected or the reaction efficiency is poor, we recommend comparing the sample with our RT-qPCR kit. As obviously the AIC mix has been optimized for COVID and optimized for using with our RT-qPCR kit. Keep in mind the Cytiva Direct RT-qPCR kit is not certified or CE IVD marked, but it can be validated by labs looking for a rapid solution for routine testing of viral samples. So that validation, you can do the validation in your lab and do your own developed LDT. Cytiva, again, has not validated the efficacy of viral inactivation with AIC mix at 70 degrees, incubation for 5 minutes. Again, labs optimizing to use our formulation would need to satisfy themselves that they have sufficiently approvals to work with infectious agents using our kit. There is a risk of contamination due to condensation on the PCR plate. We always recommend sealing after opening for the addition of the PCR reagent. We recommend using just a basic heat sealer, which I know everyone is familiar with. So just really wanted to do a quick breakdown of the products and specifications and kind of draw into some important points here. If we take a look at the full direct RT-qPCR kit, what you'll have is the AIC mix with our qPCR kit or chemistry. And we actually offer them separately. We've had customers that really wanted to just have a lyophilized ready master mix that they can have in plates or have ready to go to use with their in-house reagents. We've had customers that really just want to convert their in-house qPCR to a one-step using the Cytiva AIC mix, and we've had success with both. So we offer the Cytiva AIC mix alone if you wanted to purchase it that way. We offer our RT-qPCR master mix and reconstitution buffer separately as well, if you wanted to really just get into a lyophilized RT-qPCR. Keep in mind the RT-qPCR master mix does not include any primers or probes, but all the other necessary ingredients that are required for amplification of the target RNA during the detection step are in there. So really you just need to add your primers and probes and you're good to go. Back to the full kit itself, the full direct kit does contain the AIC mix as well as the Cytiva RT-qPCR kit. And those are all broken down. If you have any further questions, please do not hesitate to email me or give me a call. as we are winding down, I really wanted to point out to some of the products that have been supporting the COVID-19 efforts in diagnostics using RT-PCR. Just wanted to remind you, Cytiva as a whole, we are very into a variety of different aspects of the lab. Sample preparation, RNA isolation, whether you want to develop your own kit, whether you want to use kind of some of ours. I think I mentioned the Sera-extracted viral pathogen kit above. If you want to make your own kit from your own magnetic bead formulations, we offer magnetic beads as well, carboxyl and silica beads. We also offer some oligo DT beads. We do poly-A. We offer plates, spin columns. We do offer detection consumables as well. So if you wanted some nucleotides, you need a Taq polymerase or SiDye. And then back into sequencing, we have different things that you can use. If you wanted to test out for size selection or cleanup, we have our SARMAG Select. We do have T4 DNA ligase if you're really developing in-pair and like adapter ligation steps. Or if you're wanting to do some type of enrichment, we have SARMAG streptavidin beads. Then PCR, again, nucleotides. We work with kit developers, we work with end users, we work with a wide range of people. Lastly, I want to mention that we offer most of our products have the capability to be customized, and that's really where our strength lies. So we can customize many of our products, many of your in-house products. So if you wanted to make something lyostable, if you wanted to try out a particular formulation, if you wanted to do some type of contract manufacturing, if you wanted to conjugate weird things on your weird proteins on your beads or something that you have thought of that would be worked out great or would work out great, please don't hesitate to give us a call. We can help in all those aspects. Lastly, thanks so much, LabRoots, for allowing me to be here for Lab Week. Thank you, everyone who's taken the time to listen to my presentation. Again, my name is Donald Green. If you want to have more information, please reach out to me at donald.green@cytiva.com, or you can reach me at my telephone number included in the presentation as well. I hope you have a great lab week and enjoy. Thank you.
Thank you, Donald, for that outstanding presentation. We will now move into the live Q&A portion of the presentation. And as a reminder to the audience, please submit your questions via the Q&A box. Donald, our first question is, can we purchase the AIC mix separately and just use our master mix?
Yes, that's an absolutely something that you can do. We've had plenty of customers do that as well. You can use our AIC mix with any standard PCR master mix on the market.
Great, thank you. Awesome. Our second question is, can we test your Lyo-ready master mix without the AIC?
Again, another good question. Absolutely. You can definitely test our master mix if you want it. Just because it's lyostable, easy to use, we can send you some of that to test with your samples.
Getting some great questions in here from the audience. Our next question is, does the protocol take 5 minutes for all viruses?
So we can't say that is a definitive yes. What we did was we optimized the protocol for COVID-19. So there may be some optimization there that you need for other viruses. But for COVID-19, for Ebola, 5 minutes just works just fine.
Okay, great. Good to know. Moving on to our next question. Is the chemistry tolerant for all UTMs?
All UTMs except the ones that contain lysis reagents. We mentioned kind of earlier in the presentation.
Great. Another question that we just got in is, do you know if the AIC mix works with LAMP, RPA, or other isothermal chemistries?
We have not tested that, but if that's something that you want to test, we are willing to give it a try.
Awesome. And it looks like our last question here, Donald, is, can Cytiva stabilize my master mix?
Absolutely. We do have the capabilities of stabilizing your master mix. Again, it's just one of those things that you put in a request for, and we can help you out.
Awesome. Thank you for all your great insight today, Donald, and a fantastic presentation. Unfortunately, we're out of time, but don't worry, any questions that are submitted and not answered today by our speaker will be addressed via email, or for the on-demand period, those will also be addressed via email. Thank you again, Donald, and thank you to the audience for your outstanding questions. This
Thank you,
presentation will
everyone.
be available. Yep, thank you. This presentation will be available for on-demand viewing for 12 months. Please remember to share it with colleagues who might be interested in today's topic, and don't miss out on the other presentations on our agenda. Visit the agenda tab in the auditorium for a full listing. Donald, before we go, I wanted to give you the last word. Did you want to say anything to the audience?
No, I think I've said everything. Thank you for attending. I look forward to your questions. I look forward to you reaching out. Thanks.
And thanks again, Donald, and thank you to the audience for your wonderful participation. And until next time, everyone have a great day.